went back through all my notes. Which really tells me that
Okay. Well, let's go and get started for today. So if you haven't turned in your problem set, please do so right now. Come on down. It's not a big deal. Hopefully you have already. So announcements, obviously, make sure that you do Quiz 7 tonight. So we'll cover the rest of the material today for sure. And then, of course, we have our exam on Monday. Okay. So I think that's it in terms of announcements. Let's go ahead and look at a riddle. So I went into the woods and got it. I sat down to seek it. I brought it home with me because I couldn't find it. What is it? The will to live. I feel that way today even. It's been, spring break is going to be a welcome thing for me. So anyway, but no, it's not the will to live. It's something else. A tick works, yes. A tick would work, or a splinter would work. So, well, yeah. I mean, with these, you know, if it works, it works. If it explains the data, it works, and a tick explains the data. Okay, so, or a splinter. A splinter would do it as well. Okay, so, so last time we left off right here, and we are talking about glycogen metabolism, of course, and the regulation of glycogen metabolism, in particular in the liver. So, and we talked about in specific details what glucagon signaling will do specifically to glycogen breakdown. Okay, so pKa will become active. It will phosphorylate phosphorylase kinase, which will then become active, which will phosphorylate glycogen phosphorylase, which will then be active, which will then start to break down glycogen. Okay, so I would expect to see that on this assignment that you just handed in. Okay, and then if we also look at this, the other thing that would be going on that's not shown on this slide is that gluconeogenesis would be going. Right, so it does say it up here, but again, even with this problem set, pKa will lead to the phosphorylation in the liver cell of the PFK2/FBPase2 bifunctional enzyme. Okay, and in that phosphatase domain will become active. Okay, and it will start to break down fructose 2,6-bisphosphate. So you'll lose an inhibitor of gluconeogenesis. So gluconeogenesis at FBPase1 will increase in activity. That should have been shown on this. Okay, as well. In addition, pKa, and a mechanism that we didn't really talk about, will increase genetic expression of PEP-CK. Okay. PFK1 activity will go down, and finally, pKa will phosphorylate pyruvate kinase, an enzyme in glycolysis. So glycolysis flux will go down, gluconeogenesis flux will go up, and breakdown of glycogen will go up as well. Okay. So that's a quick review of what you handed in. I'll post a key of this, hopefully when I get back to my office. We'll see. If I find the will to live, I'll post it. Okay. All right. So we talked about all of those things on Wednesday. I also mentioned, and this is where we kind of left it off, I also mentioned the activity of this protein PP1C, okay, or PP1. PP1C, just that denotation C means catalytic subunit of PP1. Okay, so that is going to dephosphorylate all of these things here. Okay. It's essentially going to reverse what the glycogen phosphorylase and the glycogen phosphorylase kinase did. Now, the question though is, well, then how does PP1C become active? And we didn't address that. And so I want to go ahead and address that today and look at that. Now, key to this understanding, okay, in terms of what activates PP1C is that glucose concentrations are going to be going up when we would want PP1C to be active. Okay. If glucose concentrations in the cell are going up, then that would seem to indicate that either the liver is no longer excreting glucose at a very fast rate, or it's not utilizing glucose for its own energy needs, or a combination of the above, or blood glucose has gone up and is starting to absorb, the liver starting to absorb glucose. In all of those cases, PP1C or PP1 should become active, okay, because glucose concentrations are going up. Now, I will say this also with PP1C, okay. That protein, and I mentioned this on Wednesday, I'm going to go ahead and mention it again, is known to interact with lots of different proteins in the cell, okay, a very large number of them. And all of them are what are called either targeting or regulatory subunits of this protein phosphatase 1, okay, or PP1. And so the regulation of this can take on a variety of different quote-unquote colors depending on what cell you're looking at, depending on what process you're looking at. We're looking at liver cells. We're looking at its effect on glycogen metabolism. Okay. All right. Now let's go ahead and move on to this next slide then and think about, well, what role does that glucose play? So glucose, right, is known to, and actually I'm going to go ahead and cut this in half. We'll look at the top half here in just a second, okay? But glucose is known to interact with glycogen phosphorylase, okay? And when it binds glycogen phosphorylase, it will cause a conformational or induce a conformational change in glycogen phosphorylase, okay? And that's what we're seeing essentially right here. So we got these glucose molecules binding right here. Now, what I'm going to highlight for us here is this. There are these serine residues that, in the active form of glycogen phosphorylase, are tucked into the protein. Okay, this is not the high resolution structure of that. Okay, but they're tucked into this protein, and when glucose binds, they come out of that little hiding spot, if you will. That's what part of the conformational change does, and it exposes them, okay, to the surrounding environments. Now, what does that have to do with PP1C? Well, let's come up here. Okay, now, PP1C is this guy right here. Okay, what you're seeing down below, glycogen phosphorylase, that's this thing right here. So this is that. Okay. Glycogen phosphorylase, now it doesn't look like, in the depiction above, it doesn't look like those phosphates are tucked into the protein, but they are. Okay? So you're just going to have to use your imagination. Now, glucose will bind to glycogen phosphorylase, or glucose 6-phosphate. Either one will do this. Okay? That will lead to this, and they denote it by these red little squares to try and illustrate that there has been a conformational change. That will lead to this. Okay, those phosphates will come out. Now, that glycogen phosphorylase is in complex, as shown right here, is in complex with PP1C. Okay, and when those phosphates pop out, they become a substrate for PP1C. And PP1C will cleave those off. Okay, and that will lead to this, where we have a dissociation, what they're trying to illustrate here. This is here, okay? But they will, the two of them will dissociate upon cleavage of those phosphates. So PP1C will come off of glycogen phosphorylase. That will make PP1C active. Okay. And then it can go around, okay, active form of PP1C, can go around and start to dephosphorylate other targets, specifically glycogen synthase and glycogen phosphorylase kinase. Okay. So there's this like tiered effect of dephosphorylation. Okay. Glucose will bind phosphorylase, glycogen phosphorylase. Conformational change will occur. PP1C will remove phosphates. PP1C will let go of glycogen phosphorylase. It will then migrate to other targets like glycogen synthase and pyruvate kinase. Not pyruvate kinase. Phosphorylase kinase. Okay. And that's then describing the activities here. Okay. And it all depends on glucose concentration. Now, of course, when that happens, glycogen is made. Okay, take a UDP glucose, split off the UDP and start adding it now to a glycogen granule. Okay. So regulation of PP1C is not really straightforward. You know, it's not this like, oh, it gets phosphorylated and becomes active. Rather, there's lots of different things that have to happen to it. Okay? Questions on that? Yes? Okay, yeah, so the question is this, coming back to this. What about this? Okay, I'm glad you asked that because I was going to talk about that next. What about this? Glycogen synthase kinase, what activates that? So here's what's fun about glycogen synthase kinase. At one point in time, we thought the glycogen synthase kinase had really one target, and that was glycogen synthase. Okay? Over the last couple of decades of research, though, we have found that glycogen synthase kinase will phosphorylate. It's been demonstrated to phosphorylate over 100 different targets in the cell. It's estimated to phosphorylate over 500. We haven't discovered all of them. But that means that that enzyme is complex in its activity. And its regulation is as complex as its activity. Okay. Thank you for doing that. Shutting the door. It was nice. The hammering activity down there, you know. Anyway. Okay, so anyway. So we're going to put this, for this class, we're going to go like this. Okay? Because it's just not straightforward at all. Okay? In terms of what causes the activity of glycogen synthase kinase. Now, the other thing to recognize here is that the enzyme that we're showing it regulating, glycogen synthase, the regulation of that is not straightforward either. Because remember, there are 9 different serines that can be phosphorylated on that. 11 different kinases can phosphorylate it. Okay. We know, certainly, glycogen synthase kinase phosphorylates it, inhibits it. We know that pKa phosphorylates and inhibits it. And then there's a host of other ones that will do it as well. Okay. So we'll just leave that as an intention. Okay. And that's the way science is for sure. Other questions? That's the liver cell. Okay. All right, now for this, coming back to this, just as a, again, go back to your big picture. Okay. Elevating levels of glucose means that glycogen synthesis should occur. Okay. This is just the way it does it. Okay. Starts at phosphorylase, the phosphorylase, and then it moves from there, leading to the activity of PP1C. Okay. And then there's, you know, the biggest thing is that conformational change in phosphorylase. All right. Now let's do this. Okay. We got a couple of more slides. I don't need to. I'm not going to talk about this. So, okay. I mean, we've already talked about it. It's just insulin and what it does to glycogen synthase kinase. Okay. So it's just. Now, oh, you say, oh, good. More boxes. More green and red boxes, but you've got some purple ones in there as well. This is fun. So skeletal muscle. The other tissue type that stores a significant amount of glucose as glycogen is skeletal muscle. The liver, the main purpose of the liver in its storage of glycogen is to release glucose from glycogen and then to excrete it into the bloodstream and bring up blood sugar. Okay. In the skeletal muscle is different because the skeletal muscle is not equipped with glucose 6-phosphatase. So the release of glucose from glycogen is not going to lead to the ability of the liver to excrete glucose. Right? So the only time that the skeletal muscle would want to break down glycogen is when it needs to use glucose for its own energy needs and its own energy purposes. Of course, that would be during strenuous exercise or sustained contraction. Okay? And so that's where we essentially are going to start this conversation. We will start with this. We'll get to epinephrine and insulin okay, although we won't talk too much about insulin. But we will get to epinephrine. But let's start with contraction. Okay? For whatever reason, a muscle cell is undergoing more contraction than it would normally do. It's not at rest, it's in activity. That's going to lead to two things. One, calcium, cytosolic calcium levels are going to increase. Okay? And I'm just going to state that as just, it does. Okay? There are reasons that it does. If you've looked at the muscle contractile unit and the physiology of muscle contraction, then you probably understand why calcium is increasing. Okay? I don't want to go into that in this class, but it does. Okay? The second thing, of course, would be that ATP is being burned, which means that its breakdown product, AMP, is going up. Okay? So those are two signals that will increase in these cells. Now, let's start with calcium here. Blue. Okay? Calcium can allosterically activate phosphorylase kinase. Okay? So phosphorylase kinase, if you remember, or glycogen phosphorylase kinase, if you remember from last time, I said it's a tetramer of tetramers. Okay, and it's got these different subunits, and I said, oh, one of these is a calmodulin protein. Calmodulin proteins bind calcium. Okay? And they're a family of proteins that bind calcium. When that binds calcium, okay, in glycogen phosphorylase kinase, it activates it allosterically. Now that will then, phosphorylase kinase is going to do the exact same thing that it did in the liver, and that's going to be that it's going to phosphorylate things. Actually, some of you might have gotten confused because I drew ATP, this here. I drew this in response to calcium. I shouldn't have done that. I'm sorry. I'll give myself a red check minus. Because that would imply that it's being phosphorylated, and you're thinking, is calcium phosphorylating that? No. No, it's not. So red check minus. It won't let me give a check minus. Okay, fine. I'll go over here and get it. I'm going to erase this. I'll probably draw that back again, but okay, there we go. Sorry about that. Okay. Now, this will use ATP to do this. Okay. So glycogen phosphorylase kinase is going to phosphorylate this. Yeah. It's going to put two phosphates on it. That will activate it, and then of course that's going to break down glycogen, which will release glucose 1-phosphate, which will then be converted to glucose 6-phosphate, which will then be used in glycolytic pathway. Okay? On it goes. Now the other thing that calcium is going to do is this. Calcium is going to activate, oddly enough, this protein called calcium-dependent kinase. Okay. That is going to phosphorylate glycogen synthase. And inhibit it. Okay? And again, it can be fairly complex. But it's going to lead to overall net inhibition of glycogen synthesis. Okay? Now at the same time, AMP, okay, in addition to what's going on with activity of phosphorylase kinase, AMP will also allosterically activate glycogen phosphorylase. So it gets a double dose, glycogen phosphorylase. It gets allosterically activated by AMP, and it gets phosphorylated by phosphorylase kinase, which is being activated by calcium. Okay. Questions on that? Yes?
So when you say allosterically activates, is that after it's been phosphorylated? Because can it activate
Either.
an inhibited form of an enzyme?
Yeah. Okay, so the question, you're talking about AMP
Yeah.
and glycogen phosphorylase? So the question is that AMP allosteric activation after or before phosphorylation? Either. So there are degrees of activity that the phosphorylase can have. Okay, AMP can take glycogen phosphorylase B, allosterically activate it, convert it into essentially the A form. That A form can still be a target for phosphorylation. Okay, so or vice versa. Okay. So it can be either. Yep. Yes.
So in the presence of this calcium-dependent kinase,
Yeah.
are you showing that uses ATP to inhibit glycogen synthase A?
Okay, yeah, so the question is in regards to calcium-dependent kinase, is the ATP, is that being used to inhibit glycogen synthase A? ATP is not being used to inhibit it, but ATP is being burned to put the phosphates on it. Okay, so the kinase uses ATP as the donor of the phosphate. Okay, so to get those, to phosphorylate something, it has to have ATP. Okay, but ATP itself is not active, is not inhibiting it. Okay. It's the removal of it from ATP, putting it on the glycogen synthase that's inhibiting it. Other questions? Yeah.
Is it the same thing with glycogen synthase where you can have up to 9 phosphorylations?
Yep. Yeah, so the glycogen synthase, if you didn't hear her question, is essentially the same as what's in the liver. Okay. In regards to the phosphorylation sites, I have to be very careful with that, right? So yes, it can be as complex as 9 different serines and that kind of thing. Yes.
I see the red as these sort of dark brown. So you wrote something on one of the red things.
Okay, and you can't see it. Yeah. So which ones are you talking about? So
There's one on the far left.
this one?
Yeah.
That's glycogen synthase.
Okay.
Yep.
And then you annotated it.
Yep. It's a minus sign.
Okay, thank you.
Yep, I'm sorry. So let me put this, no, it's okay. The coloring scheme, let me put a minus here and just say that this is glycogen synthase. So if you're, let me say this too. Okay, can you read this? In the green?
Yeah.
Okay. You can read it in the green. It's essentially saying the same thing in the red. It's just going from green meaning active to red meaning inactive. Coloring is not always great. Yeah, sorry about that. Oddly enough, the shape of these proteins is not a rectangle either. I mean, that's just, I mean, I don't know if you knew that. Hopefully you took BC 401 and have a little bit more sophisticated understanding of what protein structure looks like than these blocks. They all look the same. They're just different colors. Now, this one, by the way, if you can't read this one says glycogen. So this is our glycogen granule, N+1, and this says glycogen. I'm going to need to change this figure. Okay. All right. Now, the next thing here that we need to talk about, let's go ahead and move on. Epinephrine signaling can also do some of these similar things. Epinephrine, remember, is mountain lions. Okay, flight or fight. Okay, so you get the idea. This is going to lead to the activity, again, of PKA. Now, PKA is going to do some of the same things that we talked about in the liver. Okay, so it's going to come down here, and it's going to phosphorylate this. Now we do need the ATP there. Okay, because it is going to lead its phosphorylation. Okay, that will activate it again. It can also come down here, and it can also inhibit glycogen synthase. So those are not any really different than what we talked about in the liver. Of course, the signal is different, epinephrine versus glucagon. Okay, but the activity of PKA is not different in that sense. Okay. Now here's something that is different. PKA will also, and this is not really shown here, but I'm going to go ahead and draw it out here. And I'll get to this on our next slide, but it will phosphorylate a protein called GM. Okay? It will also phosphorylate this protein that's called the inhibitor protein. Okay, so let me write this down. Inhibitor protein, sometimes called inhibitor protein 1. inhibitor protein will work for us for our purposes, okay? But that's going to phosphorylate it. pKa is going to phosphorylate it and activate it. And you say, okay, so pKa phosphorylates and activates the inhibitor. Golly, it's just so ridiculous, right? I mean, it is, but it's just the way it has to be said, though. Okay, so it's an active inhibitor protein. Okay, it also phosphorylates this GM. Now, what in the world is that all about? Okay, well, let me go to our next slide to try and explain that. Okay. The next slide here, let's look at this. Here we are. We just talked about on our last slide epinephrine. We looked at how it activates pKa, and I just said it's going to phosphorylate GM, but I didn't really describe what GM is. GM is the muscle version of the glycogen targeting protein. So in the liver there was GL, the glycogen targeting protein, but we called it GL. And it had all these things that were bound to it in the glycogen granule so that regulation could occur in all in one place. GM is the muscle version of that. Okay? Different protein, very similar name. Now when that is phosphorylated, as being depicted here, okay, PP1C will be released from that scaffolding. Okay, PP1C here, let go of. Now at the same time, pKa phosphorylates the inhibitor protein, inhibitor 1. Okay, puts this phosphate on it, active now. Of course here it's a crescent moon. On our last slide it was a rectangle. Okay, so they got, you know, they got a little bit more creative. But anyway, when that's phosphorylated and active, it will bind to and inhibit PP1C. And so I'm going to put a little negative sign here. Okay? So that when pKa is active, okay, for whatever reason, epinephrine signal being one of them, PP1C's activity goes down. Okay? Because pKa is phosphorylating GM and because it's inhibiting the inhibitor protein. I'm sorry, it's activating the inhibitor protein. Don't worry, we'll get through it. Okay? We'll get through it. Now, if it's doing that, coming back to this slide here, okay, when you look at PP1C when it's active, when PP1C is not bound by the inhibitor protein, it's going to reverse everything that was done, okay, by the kinases that we just talked about, either the calcium kinase or the calcium-dependent kinase or the pKa or the phosphorylase kinase, whatever. All of those are going to get reversed okay, when PP1C is active. Okay. So if pKa is active, then PP1C is not active. Questions? Yes?
I'm assuming it's using ATP for that phosphate group.
Yep. On the GM?
GM
And the, yes, I didn't write it down. I'll write it down. ATP, ADP, and ATP, ADP. Questions? Other questions?
The inhibitor protein is bound to PP1, then none of those.
Then none of those things can happen. Yep, so when this, right, when this is like this, these things here won't happen. Okay. Other questions?
Is there like a simpler diagram we could find?
Is there a simpler diagram? Not that includes it all. So, not all this, no. Other questions? Okay, now, here's where it becomes even more fun. And that is, well, what then activates PP1C? Yeah, oh no. It's complicated. All right, so we're not really sure. Okay, so I've looked, and I can't really nail it down for skeletal muscle. Okay, I'm not sure the field has it really nailed down. Okay. I have seen reports that PP1C can actually dephosphorylate the inhibitor protein. Okay, so yeah, question.
Would the inhibitor protein ever be active at the same time as like while insulin is present?
Okay, so the question is, would the inhibitor protein ever be active while insulin is present? And the answer, the honest answer that I have, I don't know. Yeah. So the regulation of this, in particular in the reversible direction, everything that I just showed you is pretty straightforward, at least from what I can find in the literature. The reversible aspects of it are not so straightforward. Okay, and so let me just show you this. Okay. I've seen reports that the inhibitor protein can be actually dephosphorylated by PP1C. Okay, so, which is strange because you're looking at it here and you're saying, well, the inhibitor protein, when it's phosphorylated, will inhibit PP1C. How does that work that it would then phosphorylate it or dephosphorylate the thing that's inhibiting it? I don't know. Okay, but I've seen reports that it can. Now, if that is the case, then you can imagine a scenario where as long as PKA is active, the inhibitor protein gets phosphorylated by PKA, binds PP1C, PP1C takes the phosphate off at a slower rate than PKA puts it on. Okay? So it takes it off, but then there's another phosphorylated inhibitor protein that just binds to it. And there's this kind of war between taking it on and off and on and off and on and off. I don't know if that's the case, but that is the way sometimes regulation works. Okay? Not only that, but then the GM protein, if PKA was active, would still be phosphorylated, and PP1C would not be able to access all these other targets. Okay? It wouldn't be able to bind to the GM protein. Okay. So those are more conjectures than anything else, but that gives you some maybe possibility as to what this is. Okay. And I think that I mean, specifically for the exam on Monday, that's enough. I don't want to go any further than this. We won't talk about insulin necessarily. Again, for the muscle cell, it's not necessarily straightforward. The take-home message from this would be calcium plays a very important role in this. And then, of course, PKA plays a very similar role to what we talked about, with the addition of the GM and the inhibitor protein. Okay, right? And that would then put us at the end of the material for this next exam, which puts us 15 minutes away from the end of class. So here's what I'm willing to do. I'm willing to stick around, answer questions if you've got questions for the exam, or you guys can, you know, if you don't want to hang out, get up and get out of here. Okay? I don't want to add anything more to it. Okay? We will start new material on Wednesday of next week. Okay? All right. Have a good weekend. I'll stick around and hang out if you have questions, okay?